I'm going to talk to you today about tuberculosis and how we are applying genomic tools to tuberculosis and deciphering some of the origins of tuberculosis. And I think there might be some of you in this room who came here more out of curiosity because tuberculosis, you wouldn't necessarily associate a nice shiny new building here with the study of tuberculosis. In fact, when you think sometimes about tuberculosis, some of the images come to mind are, for example, the Victorian era and innocent young maidens wasting away pale in their bed. Or maybe you think about some of the poets or some of the writers or the famous artists, the Bronte sisters, for example, whose lives were cut short by tuberculosis. If you're old enough, or if your parents or your grandparents were old enough, you might also remember the time when tuberculosis was rampant enough that we had sanatoriums that were widespread. For example, there's a famous one in New York, Saranac, and some of you might have come to. All of these images all share kind of something in common. They give you a sense that tuberculosis is a thing of the past. It's sort of something that happened in days gone by. And you might be wondering yourself, isn't really TB, isn't it cured? Isn't it over? If you think this, you're actually in good company, because not just 30 years ago in the 1970s, the medical community was predicting that TB was over. They were claiming the end, the conquest of tuberculosis, as well as other infectious diseases. Unfortunately, this is the picture of TB today. These are pictures from James Natchway, famous pictures taken of tuberculosis patients. And of course, unfortunately, it's not terribly romantic. It's definitely not a thing of the past. And it's certainly not isolated. TB is still a major killer today. In 2007, 9 million new cases of TB were recorded in the world, and it caused 1.2 million deaths worldwide.
Colored at 10,000 new cases a year?
Yes, quite a lot. Quite a lot of cases. It's quite, in 2007, I mean, it's on the low end, but yeah. This is WHO.
Australia is much less.
Yeah.
That's good.
It's good. Yeah, the Australians are doing all right. A problem still for the US. And this is particularly surprising because TB is curable. There are drugs to treat TB. We have them, we know how to treat TB, but even this is being threatened because we've seen the rise of drug-resistant TB. So the first drugs for TB were invented in 1940, and we've already seen the rise of not only resistance to one drug, but also resistance to multiple drugs, MDR-TB, and then ultimately a resistance to virtually all the drugs that are at our disposal, so-called XDR-TB, or extreme drug resistance. And some of you may remember this fellow, Andrew Speaker, who had the audacity to fly back from Europe into Canada and then cross the border into America with what was thought at the time to be XDR-TB, but what turned out to be MDR-TB. So this is a little bit grim, but the fact is it's not all bad news. There is reason to hope. There's been a resurgence of research into TB, thanks to our government, the NIH, thanks to the Gates Foundation, and thanks to many scientists who have spent a great deal of time studying TB. We have new ways of thinking about TB. And of course, the reason that we're here at the Broad talking about TB is that genomic technologies in particular are giving us new ways to think about TB. One of the ways that we can think about TB with genomic technologies, of course, is developing new drugs and new vaccines and so forth. But a surprising application of genomics is that we can use it to start to think about the origins of TB. Where did TB actually come from? And this actually gives us some insight into the disease that helps us think about how we might treat them. So I want to tell you that story, but first, before we kind of get into it, I have to give you a little bit of a primer. Namely, what is tuberculosis? Right. So the easy answer is tuberculosis is a disease caused by this rod-shaped bacterium, Mycobacterium tuberculosis. The main way you get TB is through inhaling it, and that's best shown by this illustration here out of Nature Methods. So when a person with TB coughs, they exhale many droplets that contain thousands of bacteria into the air, and those bacteria can reside for hours in the air. You breathe them in, they travel down through your lungs, reach your alveoli, and then your immune system recognizes it. At that point, your immune system calls in the first guard, these cells called macrophages. These macrophages move in to mop up TB by swallowing them. Essentially, macrophage is large eater. And when they swallow the TB end up inside these little bubbles called phagosomes. Now, for any other bacterium, this would be the end of the story. Because when a bacterium gets inside of a phagosome of a macrophage, the macrophage pumps the phagosome full of acids and enzymes that cut and burn the bacterium to pieces. And most bacteria are unable to actually withstand that. But this is actually where TB turns out to be quite remarkable. TB not only is able to survive, it's actually able to survive and persist inside of the phagosome of a macrophage. We don't fully understand how that happens. We don't really understand how TB does this, but there are at least 3 mechanisms or 3 contributing factors. The first is it turns out TB is actually able to, in some sense, commandeer the macrophage. It's able to, with chemical signaling that we haven't quite worked out, essentially block the maturation of the phagosome, so stop the acidification of the phagosome. Sort of sending orders to your macrophage, and your macrophage listens, for whatever reason. Second, TB has this waxy cell wall. So one of the characteristics of TB is it has this thick, waxy cell wall, which was part of the reason it was so difficult to actually culture TB for a long time, or at least identify it in cultures. And this protective cell wall helps TB resist whatever acidification has actually already happened. So even if it gets a little bit unpleasant in there, TB's got armor around it that allows it to survive. The third factor, or a third factor, that contributes to why TB might be able to survive is that, well, a bug's got to eat, and it's learned how to do that inside the phagosome. It turns out that in contrast to all of us, TB thrives on cholesterol. This is work by Chris Sassetti, who was the first to note that TB requires genes for essentially metabolizing cholesterol. We don't know where that cholesterol comes from. It could be cholesterol that it's already kind of packed away inside of itself, but it's also possible that that cholesterol is coming from your own cells. So in fact, it's actually possible that TB is eating your own cells from the inside out. Yes?
Those of us who have cholesterol
No, all look at this question.
drummed into our skulls that there is good cholesterol and bad cholesterol.
Yeah.
I assume so this is a form of bad?
Wouldn't it be nice if that were true? We actually don't know the answer to it. And I also wouldn't necessarily suggest this as a diet routine. You'll see in the subsequent slides, it's unpleasant. It's a little unpleasant. The side effects are bad of this diet. It's a great question. So lipid metabolism, learn how to eat it. So inside the phagosome, then TB is actually able to thrive. And it multiplies and it multiplies, and eventually TB gets so big inside, or there's so many TB inside the phagosome, it actually bursts the macrophage. Well, this is alarming to the other macrophages in the area, and they call out to the rest of the immune system for backup. Oops. Hit that one too many times. When this call goes out, the backup troops move in. A host of immune cells are pulled in through the blood supply to that area and start to form a circular structure around the infected macrophages. In a sense, these T cells, these neutrophils, these mononuclear cells circle the wagons around the TB cells. So it's a little bit like the cowboys and Indians, except with the cowboys circling the wagon, except the Indians are in the inside in this case. They're sort of circling around the TB and sort of cordoning it off. Over time, this becomes a little bit fibrous. It sort of walls it off, and you end up with a structure called a granuloma. This structure, this granuloma, is a hallmark of the tuberculosis disease. In fact, when you look at this macroscopically, a granuloma looks like a little round nodule. And the proper anatomical term for that is a tubercle, which gives rise to the modern name tuberculosis for the TB. When you're at this stage, there's essentially a stalemate between TB and the immune system. So you have the immune system cells, and they've circled the wagons around the TB, but the TB inside aren't dead. They've actually hunkered down into kind of a dormant state and sort of gone to sleep a little bit and are waiting. And that gives rise to a form of TB called latent TB. This is essentially where you have TB, but you don't know it, and your immune system isn't reacting to it, so you're asymptomatic. This form of TB has 2 major consequences. The first, it's thought that the persistent state, this kind of hunkering down and sleeping state that TB takes, may in fact be part of the reason why it's so difficult to treat TB. When TB kind of goes to sleep, a lot of our drugs don't work as well on it. And so understanding the physiology behind it is actually quite important. But the second clinical consequence of this latent TB is that it's a reservoir for active TB. It's very much an uneasy stalemate. TB inside these granulomas are essentially waiting and looking for any crack in your immune system. And when that crack comes, TB explodes out, and you get active TB. If you have a normal immune system and you have latent TB, the chances that you get active TB are about 10% in your lifetime, roughly 10%, more or less. If you're immune compromised, though, if you have AIDS, that goes up to 5 to 10% per year, partly explaining the tremendous synergy that we're seeing between HIV and TB throughout the world. The scary statistic, then, is that 1/3 of the world appears to harbor latent TB. Now, to scare people, you say 1/3 of this room. It's actually not 1/3 of this room. It's most of the people in Africa, most of the people in Russia. But it's low.
You mentioned in the United States.
Yeah, the United States is low. It's very low. But we're probably good. There's going to be a few, though. There's going to be a few people here with latent TB.
Well, means low 5%?
Yes, it's low. But it depends on where the people in this world are from. If you have people here who may have come recently from Africa, from China, from Russia, if you came from a Russian prison recently, you're really in bad shape. For a lot of reasons. Yes. But this is a reservoir. The point is that 1/3 of the world, wherever they reside, is a reservoir for TB, just waiting to erupt onto the scene. Right? So when TB goes active, then the TB spill out, and if it's left unchecked, two rather undesirable things happen. The first is your lung starts to become consumed. Effectively, as TB wars with your immune system, it produces these cavities and these necrotic centers in your lung that cease to function, and your lung slowly ceases to be able to work. And that's number one. Second, as a consequence of this tissue degradation, and also as a consequence of the fact that your immune system is amped up because you have active TB, you also see this characteristic wasting. TB patients look like they're sort of being consumed from the inside out and lose body mass. Finally, when you have active TB, these free mycobacteria will occasionally gain access to your airways, you'll cough them up, and that starts the cycle again. So it's worth stepping back for a second and marveling at this. This is actually a remarkably complicated disease mechanism, right? This is not for bacteria that are faint of heart. This bacterium is literally marching in to the heart of your immune system and setting up camp and managing to do it, and do it well. So one of the obvious questions, and one of the big questions in TB scientific research, is how does it do it? This is not for the amateur bacteria. The scientists who study TB use the idea or the term professional pathogen. TB is essentially a professional pathogen. It does this for a living, and it does it well. The question is, how did it become a professional? How did it go from being a rank amateur bacterium who got knocked around by macrophages to being a professional bacterium that could actually start to commandeer these macrophages? That's partly at the heart of us trying to understand TB. And one of the interesting ways that we can get at this question is to start to ask, when did TB arise? How did TB become the disease we know? In effect, we want to go back and look essentially at the evolutionary transcript of all of TB's training throughout the years to try to say, how did it get to the point that TB was able to do what it does to us? Do I? Can you hear me? You know what? Excellent, all right. Everybody hear me? Fantastic. So that's the story we want to tell. We want to get to the point, we want to ask the question, where did TB come from? And it turns out that in the last 3 to 5 years, we've made tremendous progress. It's really quite exciting. We're able to put together quite an interesting picture of where TB came from. It involves using genomic technologies, and we're going to get to that. But to start, and because it's just kind of a convenient place to start, we're not going to start at the beginning of TB's history, but rather we're going to start at the beginning of our history with TB. So this is a very famous quote. It's actually several quotes kind of stitched together. And the reason it's famous is not just because it's accurate. It actually is fairly accurate description of a disease we would call TB, but rather that it was written a long, long time ago, about 16 years ago, by the guy you'd expect to be writing this sort of thing, right? And in fact, there are many, many examples in the ancient written texts of apparent descriptions of TB. We see examples in the writings of Homer. There are 2 mentions of it in the Old Testament from the time that the Israelis were captive in Egypt. There are a number of descriptions of it in the Indian Vedas, as well as some discussion about how to treat it or whether to even treat it or not, as well as discussions in Chinese medical text. They take us back quite a distance But I say apparent because, of course, in all these texts, they didn't say tuberculosis. Right? Nobody mentioned tuberculosis. For one thing, they didn't speak English. They were writing in their own language. Right? But more importantly, they didn't know about, I mean, we talked about the tubercle, right? At this time, they didn't actually know. They hadn't put together the link between tubercles, which they might have seen in animals, and a disease that we would call tuberculosis. So instead, they described the disease by the symptoms that they saw in people who had the disease. So for example, Hippocrates used the term phthisis as his description of this disease. And this roughly translated in Greek means a wasting away, more or less the description of the symptoms. A similar description was picked up by the Romans in Latin, consumere, which then went through French and Old English, and eventually came to the word that got used for most of the history of TB, consumption, which is once again a description of the symptoms of TB rather than the disease pathology. But these aren't the only two words. There have been many, many terms that have been used to describe TB in different languages. My favorite here, I don't know how to pronounce this, but it essentially means the waning moon, very descriptive of the course of TB, right? Very interesting. The reason why this is relevant is that because there were so many terms used for a disease we think is tuberculosis, and because they were describing symptoms, it's hard to often know for sure they were talking about tuberculosis. I mean, there are many diseases that can cause these symptoms. Cancer can cause wasting away. Pneumonias will cause the kind of coughing that you see with TB. Fortunately for us, TB leaves a sign. So I've described TB as primarily a pulmonary disease, and that's true. But in fact, TB infects many more organs besides the lungs. It can affect your brains, lymphatics, pleura, other inconvenient places. But the infection that's relevant to thinking about the origins of TB is the fact that TB can infect your spine. When TB does this, it's called Pott's disease, named after the 17th century physician who first described it or sort of named it and knew it was TB. And when it does it, it tends to go, when TB infects your spine, it tends to head towards your thoracic vertebrae, although it'll hit all of them, and heads directly for your vertebral body and hunkers down and causes an inflammation right here in the anterior aspect of your vertebral body. Over time, this inflammation will cause a weakening of that interior body. And if you imagine your spinal cord is sort of a stack of these vertebral bodies, and if one of them starts to give, you can sort of see what would happen. It collapses, clicks on top of itself. This is a very severe form of kyphosis that arose because of spinal TB. This, of course, gives rise to hunchback. Hunchbackism. Kyphosis is just a fancy term for being a hunchback. It doesn't necessarily kill you. It's unpleasant, but it doesn't kill you. This kyphosis gives us an archaeological signature that we can go and look for to look for signs of TB. And this has been done extensively now. The most famous examples of this were actually back in Egypt, where people went to Egypt and looked at mummies and identified numerous cases now of mummies that had clear signatures of kyphosis, as well as a certain number of other anatomical signatures of TB that we can identify in skeletons. This fellow right here was the high priest of Amun, and this was about 1000 BC. But even this isn't definitive, because this is still, I mean, there's lots of things that can cause this kind of kyphosis. The great thing about the fact that of TB going to the bones, though, is that as much as the bones protect themselves over time, they also protect whatever's in the bone. And we can go into some of these skeletons now and actually pull out molecular evidence that TB was there. We can pull out pieces of DNA that suggest TB and actually tell what kind of TB, and we can also pull out molecules here that are sort of diagnostic of TB being there. So, oh yes, and then finally, when we looked across, when people have looked across large numbers of different sites and different mummies from the pre-dynastic all the way through to the end, we see high instances of apparent TB. 25% of mummies in some studies look like they had TB. So really high instance of it.
You mean Egyptian mummies?
Yes, Egyptian mummies. Yeah, we'll get to some of the other ones. But there are. So let's put this together now. So what is this telling us? When we take all this data and kind of stick it together, we see a very interesting picture. If we just look at the data that we've talked about so far, and then we throw in the fact that there are examples of fossils here that from Britain at around 400 AD, interestingly enough, we see a picture where already by the time of the city-states, Egypt, Sumeria, out to the Roman Empire here, TB was present, and not only present, but if we believe the 25% in mummies and descriptions of Hippocrates, it was present and rampant. And we'd expect that this could be possible because city-states have reasonable populations that can harbor infectious disease. But if we look at the fossil record more closely, it actually pushes us back further. Fossils from Italy, Germany, and then finally the oldest known fossil of TB. And this is actually not just a fossil specimen, but a specimen that's also been molecularly confirmed. So we actually have DNA now and mycolic acid. Puts us all the way back to 7,000 BC, where we have evidence of TB. This was actually off the coast of Haifa in a submerged site. So this is pretty striking, because now this is before the city-states now. This is all the way back, essentially almost to the point of the agricultural revolution. And this has several interesting consequences when we look at this data. The first, and I'm just going to put it out there, you note that there are no fossils over here, and for a long time people assumed that, like smallpox, measles, et cetera, TB got to the Americas with the Europeans. Let's sort of leave that there for a second. But in addition, the fact that TB takes us all the way back to the agricultural revolution gave rise to one of the oldest, I would say, theories about where TB might have come from. Namely, since the agricultural revolution gave rise to the domestication of animals, the theory was that we must have got it from animals. TB must have come from animals. And this, of course, has precedent, right? We're all very familiar with the fact that flu might come from ducks and birds and pigs, but also the common cold presumably might have come from a horse. Measles might have come from a rinderpest from the cow. In the case of TB, the prime suspect was the cow. And the reason for this is that tuberculosis isn't just a disease of humans, it's actually also a disease of animals, including the cow. And it's not just the cow, it's the seal, the goat, the oryx. Many other species get TB. These four are interesting, though, because they all get TB from a type of mycobacterium called Mycobacterium bovis. So this is a TB that's kind of similar to the TB that gives us a disease. And the fact that such a thing exists gave rise to this famous hypothesis. Human TB might have come from cow. And I phrase it as a question because it's still a little bit of a question for this talk. Did human tuberculosis come from cow? And in this case, it would be M. bovis being transmitted to M. tuberculosis. If this were true, this would have interesting consequences because, first of all, it postulates that TB is a fairly recent jump from animal to cow. This is consistent with some molecular data, but also, when we think about how long it might have taken for TB to become the professional pathogen that it is, it's a little bit surprising this could be so recent, right? So there's some reason now to be a little bit suspicious. But more than that, the fossil record starts to give us some sense that maybe things aren't quite all right. Okay? First, let me just go back to this question of what happened in the Americas. Well, we now know that TB was present in the Americas before Columbus. Absolutely. We have mummies, Peruvian mummies in this case, that have been identified molecularly now with TB long before the Europeans made it over to the Americas. But most strikingly, somewhat recently, a last piece of fossil evidence was discovered in Wyoming, in this case in a bison that had fallen into a pit along a game trail. And it turned out this bison, molecularly confirmed, had M. tuberculosis. Not M. bovis, but M. tuberculosis. 17,000 years ago. Well before we were domesticating animals or cultivating plants. We were hunter-gatherers at this point. And yet we see M. tuberculosis.
Were there any humans in North America at that time?
There were, absolutely. So the Bering Strait carried some humans across. And keep that in mind, because that actually is an important clue. Right? How did this happen? How did this get there? Right? But to actually answer the question requires that we have better tools than fossils now. So we have to be able to look deeper in time. We have to go further back in time than the fossil record necessarily can easily tell us. And that's where we get to genomics. Genomics is giving us a tool to look further in time. So in order to understand that, I'm going to have to give you a little bit of a primer, because I don't know to what extent folks here are familiar with bacterial genetics, but let me just kind of give you sort of a quick primer of what bacterial genomics allows us to do. So what makes a bacterium work, essentially, is that inside its cell wall, and its plasma membrane, there exists a loop of a substance called DNA. I think you guys are probably all familiar with DNA. It's genome sequence, right? And as you're undoubtedly all familiar, its genome sequence is literally just a string of A, T, Gs, and Cs. The interesting part about this loop of A, T, Gs, and Cs is that there are segments of the genome that we call genes that when they are essentially turned on, give rise to first a molecule called mRNA, but then ultimately a protein. And it's these proteins that go on and do really useful things. They go on and, for example, do metabolism and maybe take cholesterol and turn it to energy, for example. So this is really very basic view. What genomics is allowing us to do is to essentially get the readout of this complete string of A, T, Gs, and Cs. It's effectively letting us get sort of the parts list of a bacterium, because that's what that is. Each one of these genes codes a protein. The proteins are really the parts of the cell. It's what makes the cell do what it does. So if we get this DNA, we get those parts. The human genome, of course, is the most famous example of this. Your parts list is now known. It's out there, 3 gigabases. The parts list for Mycobacterium was also deciphered in 1998. And this is a picture of it. It's a circular genome. It's a lot smaller than yours. It's about 4 million bases as opposed to 3 billion bases. And it has about 3,000 genes. Each one of these little arrows here represents a gene. If we kind of zoom in, we can see a little bit how they're spread out. This is the parts list of TB. And it's already giving us some interesting insights. So I've zoomed in on this region in particular because where you see these light blue boxes above these red, the red are genes, and the light blue boxes are an annotation on that gene. This light blue boxes is an annotation that's telling us the genes underneath are involved in lipid metabolism, i.e. cholesterol and other lipids. In fact, one of the interesting surprises to come out of the TB genome was that there was a host of genes, a huge number of genes for lipid metabolism. Makes some sense based on sort of what we know about the genome, or about the pathology of TB. But it's giving us a hook now. We have some of the parts that might be responsible for how TB is living off of cholesterol, for example, and also making its cell wall, because the cell wall is made of lipids as well. But we can go further than that. We can go further than that. And to go further than that, we have to fill in this picture, because I left out one important thing. I said when a gene gets turned on, it produces a protein. But the obvious question is, how do you turn on a gene? Right? How do genes get turned on? It turns out genes are turned on by other proteins. So when a gene converts to a particular type of protein called a transcription factor, that protein binds to kind of a switch on a genome, your genome, some other genome. Think of it as a switch, sort of analogous to a switch. It flips that switch, and this second gene turns on, and maybe it codes for a protein that does something else. You can think about this as kind of a circuit. This is the analogy, a metaphor would be a circuit, because what you have is protein 1, when it goes on, it turns protein 2 on. That's like a little bit of a circuit. You flip a switch on in your kitchen, a light goes on somewhere else. That's a circuit. Bad circuit, but it's a circuit. We can, with the current genomic technologies, we're actually able to start to determine the wiring diagram of this circuitry. I can't go into the details of it, but let me give you a sort of motivation why this is useful. So let's imagine you're trying to understand this home stereo system, right, which looks nothing like mine. But if a repair person came in to look at this home stereo system, one of the first things they might do, in fact, is go look at the back, turn it around, and look at the wiring diagram. Because the wiring diagram tells you a lot, right? It tells you, for example, what's connected to what. And also, if you look at this carefully, you didn't really know what was going on here, it could also start to tell you what's really important. For example, if you knock out the antenna, yeah, sure, you're not going to get over-the-air digital TV, but really, who watches over-the-air anymore? But absolutely, your DVD will still work, your tuner will still work, all the rest, most of the parts of your system will still work. But if you knock out your receiver, God forbid, the whole thing is shot. Right? We would call your receiver kind of the hub of your home theater system. It's sort of at the heart of your home theater system. A similar idea actually applies to bacterium and TB. This is a wiring diagram for part of TB. This is going to be, it's a little bit hard to interpret. Let me just sort of walk you through it. The basic idea here is this is not all of TB. This is just the part of TB responsible for fatty acid biosynthesis. The red dots are genes that are turning on other dots, which are in yellow, and the line just indicates a wire. So when a red dot is connected to a yellow dot, it means that that red dot is turning on that yellow dot at some point during the life cycle of TB. This is a circuit diagram, in effect, for TB. And if you notice and look carefully, this little red dot here looks a little bit different than some of the others, right? It's wired up to a whole lot of stuff. This red dot is wired up to all these little yellow dots. This is a hub in the circuitry of TB. And this hub, it turns out, is responsible for fatty acid biosynthesis. We're going to return to this point in a second. But we're doing this for all of TB now. That's a really exciting thing that we can actually build this circuit diagram. This will have relevance to the orders. We're going to get back to that. The last thing that genomics is allowing us to do is that it's actually allowing us to put together family histories. We don't have to just sequence one bacterium. We can sequence hundreds now. The technology has scaled up such that sequencing 4 million base pairs is easy. We can go out now, instead of just taking one TB, we can have a collaborator give us literally hundreds of TB, and we can sequence them in a short order. That allows us essentially to take each one of those sequences and build a tree. Let me try to motivate how we would do that. You've all seen evolutionary trees, right? So this is an example of our evolutionary tree. And the basic idea here is that it's saying that human and chimps are more similar or shared an ancestor more recently than, say, that ancestor and gorillas and so forth. If you were to do this using sort of old fossil records, the way you go about doing this is you look at a fossil of chimp, you look at a fossil of humans, and you sort of say, roughly speaking, well, there are a lot of similarities, therefore they must have shared an ancestor fairly recent, and they're less similar than, say, the tarsier. Right? That's exactly what we do with genome sequence. So if you just replace these organisms now with genome sequences, all we're doing is taking genome sequences and asking, how similar do they look, and we build a tree. And that gives us a family tree. A useful thing about this is that if you build this tree right and you have the right tools, you can start to get time out of it. Because a family tree is looking back in time. As shown here, it's allowing you to guesstimate, and they're very rough estimates here, of when did these ancestors arise. So that, for example, the ancestor of the New World monkeys and all the rest of these guys occurred somewhere between 40 and 50 million years ago. It gives us time. We can now do this for TB. And the first attempt to do this for TB is shown here. We don't have to get into the details. I just want to kind of give you a flavor of what we see, and then we're going to explain it. This was work done by Sebastian Gagnon at MRC. And what he found was when he took a large number of M. tuberculosis strains, and really hundreds, he didn't sequence their whole genome. He just sequenced some of their genes. He found something very interesting. TB aren't all alike. We wouldn't expect that. But interestingly, the TB fall into these different groups on the tree. It's like little small sort of subclades within your family. The Johnson branch or the Robinson branch of your family, for example. And what was interesting about this was that these little groups also happen to come from different parts of the world. How do we explain this? This brings us back to our origins. So don't worry if you don't completely understand it. We're going to explain this. And you're going to see that the explanation is very, very satisfying. Just note, there are these groups, right? OK, so we have genomics. It's letting us go back now and revisit the history of TB and start to think of and ask the question, where did TB come from? And we're going to answer this question, but remember some of the mysteries. TB has been evolving. Well, we think TB must have been evolving as a pathogen in human for a long time, and yet we have this hypothesis that TB came from cow, which was fairly recent. So that's one interesting tension. We're going to tell the story in a couple of chapters, short ones, and then I'm going to summarize at the end with one diagram that will sort of make it all become clear. So, but we're going to do this in just a few chapters so we can kind of see how we answer the various kind of puzzles that have come out of this work. To start with, we're going to start just by thinking of all the TB as one family, and instead of looking at that family, we're going to look back in time at all the relatives that this family had. So it's a little bit like taking your family tree, and instead of thinking about your brothers and your sisters and so forth, it's going back to your ancestors and asking who was your late ancestor. If we do this, we find something rather remarkable. A large number of the ancestors of TB came from soil. They were soil bacterium. Now, to some extent, this isn't hugely surprising. There are a lot of bacteria in soil, and occasionally bacterium in soil will hop and go someplace else. But these particular bacterium are telling us something actually very interesting potentially about TB's pathology. This one in particular, Rotococcus, which is a very ancient relative of TB, going all the way back here, I'll tell you how old in a second, turned out to be particularly illuminating. So this is a bacterium that's been studied widely because it's being used for biofuels. And the reason is it happens to be a professional scavenger of organic materials. It can digest a wide range of just about anything. And in particular, recently, it can digest pollutants. As we've polluted the atmosphere, Rotococcus goes and digests these pollutants. It has a neat trick, which that it takes these organic materials, converts them into lipids, sticks them into a little bubble inside of itself, and then saves that for a rainy day. And when it gets hungry, it eats those lipids. Very useful trick. It fattens itself up, essentially. But the interesting thing is that one of the nutrients that this organism can digest is cholesterol, it turns out. And when we compare the TB genome with the Rotococcus genome, so these are like, they're two species that are very, very distantly related, a surprising thing pops out. It turns out that the genes, the parts for doing this cholesterol metabolism are the same, more or less. There's some differences, but they're basically the same. Not only are the genes the same, but some of the switches, in fact, virtually all the switches that are in Rotococcus also exist in TB. It's like taking two houses that were on two different continents and going and looking at their wiring diagram, and sure enough, they had the exact same wiring diagram. Really remarkable. Which circuit is this? This is exactly this hub that I showed you on the slide earlier. This hub, which is so critically important for cholesterol metabolism, it turns out arose 1 billion years ago, the B. Long time ago, in soil, this circuitry for digesting cholesterol looks like it arose there and then was actually conserved not just out to tuberculosis as it became a professional pathogen, but also out to many of these other, in fact, most of the other organisms that we've looked at, with a few minor exceptions that look funny. This circuitry looks important. So this is actually surprising because now it's telling us, well, when we think about how TB learned how to digest cholesterol, evidently, it did this about 1 billion years ago. This is roughly speaking, of course, because it was an ancestor of it that acquired these genes. But they were important enough that over the ensuing billion years, those genes were conserved as kind of a genetic gift, if you will, to subsequent generations until TB became a professional pathogen. So that brings us to then the next step in the evolutionary chain is, well, when did that happen? When did TB become a professional pathogen, right? So we're in soil here, and yes, we can digest cholesterol, and this is a very useful trick. When did this trick get applied to us as dinner? Right? To do that, we now have to turn our attention back into the family, into TB, into that family. And the answer here, or at least an answer, our best understanding of it was actually put together by Cristina Gutierrez here at the Institut Pasteur, fantastic scientist and a fantastic story. Her trick was to look at members of M. tuberculosis and then take some close cousins, M. cannedii and M. africanum, who are also human pathogens, and compare them. The rationale was, if these all three are human pathogens, maybe she could triangulate back in time to the ancestor of all three, and that would tell us when pathogenicity arose in humans, right? So she's going to take all three of these things and go back in time. And sure enough, she could do it. I mean, it's not terribly difficult to do. But the answer was really surprising. What she learned from this was really surprising. She learned two things. First, because cannedii and africanum are pathogens in Africa, the parsimonious explanation for this tree is that pathogenesis in humans I put in quotes, arose somewhere in Africa. This is not terribly surprising if you think of our own history. What is surprising, though, is when she uses this molecular clock to date when this happened. It happened 3 million years ago. 3 million years ago. So if you think about this theory that TB might be recent, say 10,000 years ago, This completely shoots that out of the water. This is actually telling us that TB became a pathogen of us before we became us. It started practicing on our ancestors, and it's been practicing for 3 million years. That's a lot of practice. And it did a good job. 3 million years old. Now, I want to say this is still up for debate, but this is looking very solid. And consistent with this is the fact that very recently, and this is even more up for debate, a fossil was unearthed in Turkey of Homo erectus, which was about 500,000 years old, that looked like it had TB of the spine, or TB of some of its bones. No molecular evidence necessarily, but TB of the bones. This is fairly contentious, but it's at least consistent. So now, of course, what about cow, right? Poor cow has sort of been the prime suspect for TB all these years. Well, what happened to cow? What's going on here? And this linked to 10,000 years ago. This was finally put to rest by Stuart Cole, actually quite independently of Christina, although we're sort of putting it on the same tree. And what he found was actually really quite interesting. It turns out that contrary to TB in humans arising from cow, it was actually the other way around. Yeah. It had this bum rap all this time, and in fact, it was us, more or less. TB, bovis, in fact, was a strain that arose out of this M. africanum strain, so presumably somewhere in Africa. And it arose about 70,000 years ago. That's roughly speaking. So this is it. Wasn't cow to human, but rather a hominid, because we weren't humans necessarily back then, through to all of these species. There's one last chapter, and then we're going to summarize it. And this is kind of a short chapter because we have to summarize it all for this chapter to make sense. We go back to Sebastian's data now, and you see this tree, and it has all these different groups. And the question is, can we make sense of all these groups now in the context of what I've shown you so far? It turns out we can. It's actually pretty easy. This group of organisms here, which are ancient organisms, those actually all correspond to M. africanum and bovis. So ancient here is low down here. That only leaves the modern TB to explain. And there are 3 groups here in the modern TB, purple, blue, and red. 1, 2, 3, that we more or less have to explain. Okay, 3 different groups. How do we fit those into this tree? Well, it turns out it's fairly easy. The modern TB are, in fact, these guys. And this tree here is telling us that at some point, there was a split from this ancestral lineage, and one lucky TB got out and split into 3. And by using a molecular clock and some other evidence, we can guesstimate now. It's just a guess, but it's still a reasonable one, that this happened about 35,000 years ago. Now, I don't know if this number means anything to you, but what's really unique about this number is that this is very close to the time that man, us, modern man, was leaving Africa to colonize the world. What this is suggesting is that TB got to all these places on the back of us as we left Africa about 35,000 years ago. Okay, let's put this all together now. That was 3 chapters that were fairly complex. Let's put this all together now in one story that will give us a clue, because there's still one mystery. How did TB, after we left Africa, get to all these different places? And Sebastian has a very nice answer for that. So let's put this all together now in a story and summarize what we've learned. If we summarize what we've learned, we know the story starts back about 1 billion years ago. 1 billion years ago, some ancestor in soil, long time ago, acquired the ability to catabolize cholesterol. And that was deemed useful enough, it was useful enough, that descendants of this soil bacterium maintained it over the subsequent millions and millions of years until about 3 million years ago, one lucky descendant decided to try and invade us and successfully did so into one of our hominid ancestors. Over the next thousands and thousands of years, this descendant evolved with us. M. prototuberculosis is what that descendant was, we call now. And sometime before 3500, what we would call M. tuberculosis arose somewhere here in Africa. In the meantime, a whole completely different descendant of M. tuberculosis gave rise to the strain that infects cows, and cow is now exonerated. And then there were a couple of early migrations. M. africanum went out here to Africa, out to West Africa, and then there was an early migration of man out along the southern route here, presumably overland. There's some evidence that suggests that man went overland south into the Asian islands early on that brought some of TB. But the biggest road trip, of course, was when we left Africa and brought TB with us. We first went to the Fertile Crescent, and from there spread to various parts in the world, including Western Europe, China, and India. Presumably, at this point, although we can't be certain, and you asked this question, some lucky group of people, and therefore some lucky group of TB, crossed the Bering Land Bridge and made it over into the Americas, thus giving rise to our bison fossil. And then this is really speculative, because we don't know this for sure. Presumably, TB came down here through Mesoamerica into South America. I say this is speculative for the following reason. We absolutely have no fossil record in Mesoamerica of TB, of ancient TB. For whatever reason, there's a lot of fossils, and none of them seem to have signs of TB. We get them in Peru, we now have them up there, but right here, it's a little bit of a mystery, kind of a hole. Back in Europe, ancient civilizations arose, gave rise to the artifacts and the fossils that we talked about previously. The last step in the story of TB was beautifully summarized by Sebastian Gagnon in his paper. How do we get from this to that picture of TB in various different parts of the world? It happened effectively in two steps. First, there was a population explosion. We went from hundreds of millions to billions of people over time, from the fall of Rome, say, through to especially the Industrial Revolution and then up through us right now. And this happened, of course, everywhere, but in particular, it happened most extensively in Western Europe, India, and China. And the TB that were lucky enough to be present in these booming places also boomed because TB grows on us. And then with world trade, those TB once again hitchhiked on us. And the ones in China went to parts of Asia. The ones in India came back into East Africa, and the ones in Europe went just about everywhere. And this gives us our picture of modern TB. This is the story. This is effectively the story of TB from 1 billion years to today. This is the story. It's a story. So let me just throw a caveat out there. This is a story. There's a lot of holes still. There are a lot of things that we want to ask, and there's still a lot of fuzziness here. Obviously, from 1 billion to 3 billion, 3 million, that's a lot of time. And we want to kind of fill in some of those gaps. We're actually doing that. So folks in my group are going and looking at the fossil record, but the molecular record, and trying to fill in the gap and ask, how did the circuitry of cholesterol metabolism evolve from 1 billion years to 3 million and to today? And with our genomic tools, we're actually able to do that. In fact, the way sequencing is going now, with the cost going down every year, soon we'll be sequencing thousands of TB strains and related strains. And this is going to give us an unparalleled picture in terms of refinement of the evolutionary history of TB. And of course, there are many other questions. What happened in Peru? How did TB get there, et cetera, et cetera. The point is not that this is the exact right story, although we think it's pretty good. I think it's pretty good. Some holes, it's pretty good. The point is that we're actually in a position now to even be able to think about doing this. That's what's really exciting. And it's not just an academic exercise. What we found by looking back 1 billion years might actually tell us something about how TB causes disease today. Yes?
Do they think those genes were laterally transferred or vertically transferred?
It looks vertically transferred. It's a great question. It's a great question. Yeah, so since you asked that question, one thing that I'll say is that because when we go back 1 billion years, there's lots of lateral transfer. But once you get into M. tuberculosis, there's actually no lateral transfer at all. It's one of the interesting things about MTB. It's clonal. And it seems to reflect the fact that it became a pathogen in humans and started to lack the opportunity to have lateral transfer. Great question. Yeah.
Does it seem like the disease has gotten more or less virulent? So 25% of the population of Egypt had TB. They weren't all sort of dropping dead of TB, right? Does that mean it was less virulent than it is now, or?
But there were a lot of people dropping dead. Death was happening, for sure. And in fact, if you go to, say, the 1700s or the 1800s, by the 1800s, 25% of all deaths were as a cause of TB, or as a consequence of TB. So do we have any evidence to suggest that over the longer historical period, TB was more or less virulent? We don't, but it doesn't mean that isn't a possibility. There is suspicion that there may be strains now that might be more successful than others. There's a very famous strain called the Beijing strain, which seems to have been a little bit more successful than many of the other current TB strains. And there's some suggestion that maybe it gives rise to more drug resistance than others. It's still very kind of iffy, but there's a possibility that there are strains out there that are more, certainly different than others. Yes. You know, sorry, I've been asked also to have the microphone passed around if we're going to have questions, because they're recording this. Oops, sorry. Yeah. There you go. Yeah.
A question about the latent TB. And,
yes.
Is there any biomarkers for latent TB, and is it possible to cure it at that stage?
That's a great question. So 2 great questions. People are absolutely looking at biomarkers. So a collaborator of ours at the Max Planck, for example, is looking to see whether or not using mass spec we can detect chemicals in your urine, for example, that might differentiate between latent and active and so forth. And I can tell you that they have some promising results, for sure. Can you cure latent TB? Yes, you can. So there are treatments for latent TB. It require, the sort of standard treatment is isoniazid treatment over a long period of time, say 9 months of isoniazid treatment, but people are looking at other treatments as well that might shorten that period. So you can actually be cured of latent TB. Although it's a little bit difficult to know for sure that you've been cured of latent TB, because it's just. Can you predict latent TB? Yeah, so that's where the biomarkers can you predict? Not yet. Yes, ma'am.
Since the cholesterol metabolism circuit is over 1 billion years old, what was the sources for the cholesterol that it was working on?
That's a really great question. Probably plants. So the suggestion is that, so sterol rings, which are included in cholesterol but also in other chemicals, are frequently produced by plants, sometimes as a defensive mechanism. And so the hypothesis would be that some of these molecules that have sterol rings might have been derived from plants, possibly as a defense. And well, not Rotococcus, but an ancestor of Rotococcus learned how to defend itself against that. A great question.
Are there any similarities to how we metabolize cholesterol?
Yeah, so that's being worked out as we speak. We don't get energy from it in the same way that bacteria get energy. So there are differences. There are definitely differences there. But Chris Sassetti is the right person to ask about that. I mean, I don't think it's known right now. It's a great question. Yes?
Do you foresee like one treatment being available to all types of tuberculosis all around the world? Meaning, is there a difference between tuberculosis strains from Africa and America right now?
Yeah, so that's great, a great question. I guess I would rephrase that and say, do we see the need right now to have different treatments? And I don't know that we necessarily see the need right now. One of the reasons Sebastian's looking at this diversity is to try to ask the question, does this diversity actually have a physiological difference? Right now, the driver for different treatments is actually drug resistance. And so our current treatments for drug-sensitive TB are actually effective regardless of where you apply them, and that's been well documented. It's when you get drug-resistant TB that things start to get a little bit difficult, and you have to have sort of secondary and tertiary strategies for treatment. Yes, ma'am.
Are there drug-resistant vaccines as well?
So there is a vaccine for TB. There's a vaccine for TB. It's actually derived from M. bovis. But you're asking, is there a vaccine that will protect against drug-resistant TB?
Is there a drug? Are they finding vaccines are working anywhere?
Yeah. So the BCG vaccine, it turns out, isn't terribly effective.
Is that the gold standard?
That is the gold standard. It's the only one we've got.
So it's waning.
It has waned a long time ago. It never actually was all that strong. So it's been determined that the thing that BCG vaccine is particularly good for is to protect children against the most severe side effects of TB, particularly meningeal TB. But by the time you reach adulthood, your immunity is worn off. So for example, I was BCG vaccinated because I was born in Hong Kong. And at this point, you would have my immune system has absolutely no memory of ever seeing that. And that would be true for lots of people here who've been BCG vaccinated. It just doesn't seem to hang on. One of the interesting, just since you asked this, that the challenge with vaccination to TB is that how do you prime the immune system to get rid of an organism when giving it the entire organism isn't enough to prime it, right? TB is actually already in you. And you give all of TB to you, and it's still not enough for your immune system to kind of say, okay, good, I'm going to knock it out. The answer might be timing. It might be that you have to prime the immune system before it sees the whole TB. I mean, that might be an answer, but this is not, I'm not an expert there.
So with like the increased international travel that we have, people becoming exposed to the disease, becoming latent carriers, do you recommend routine screening for TB? And if you do see, or they do come up, say, positive with the tuberculosis skin test, how cost-effective would treatment be at that point if they're not exhibiting any symptoms? And then can that actually progress to, say, another epidemic? What's your view on that?
Yeah, this is great. So I'm not a clinician, so I want to be sure that these aren't seen as advice in that sense. But based on the people who come up with these recommendations, routine screening is primarily needed when you're working in an environment where encountering TB is really going to be an issue. So if you're a nurse, if you work in a homeless shelter, if you have to work in a prison, then this becomes a critical thing. So if you want to go volunteer in a hospital, you'll get TB screened, for example. Otherwise, it's not particularly necessary. Then your second question is, if it spreads, can it give rise to an epidemic? Well, personally, I think the danger is not an epidemic of TB, because when TB started to rise up in, say, New York in the 70s, once we knew it was happening, we could clamp down and we could stop it. So it's not TB that's the issue. It's really drug-resistant TB. That's the real scare. That's what could cause an epidemic. Because if there was an outbreak, a massive outbreak of drug-resistant TB, and it was resistant to the drugs that we use routinely, that really could cause a problem. And of course, it is causing a problem in some countries where they're not able to treat drug-resistant TB because they don't have access to the drugs.
So let's take a moment to thank James. I'm sure he'll be